Hi there. So this is the digital version of a talk that I recently gave at the Southeastern Geological Society of America meeting that took place this past week in April 2018 in Knoxville, Tennessee. And my talk is focused on looking at some very unusual preservation of Silurian age fossils called holocystids. And these holocystids belong to a particular group of echinoderms, the relatives of sea stars and sea urchins. And these unusually preserved fossils have some incredibly important implications for understanding these evolutionary trends. So with that, my colleague and I, Dr. Colin Summerall, would like to thank you for taking a few minutes to listen to this talk. All right, so echinoderm taphonomy. And taphonomy is the science behind understanding what happens after an organism dies to when it's found as a fossil. So with echinoderms, their taphonomy is quite interesting. So we take a look at this beautiful starfish, right? Absolutely gorgeous fossil. This is called Astropecten. You'll notice the names of these fossils are found in the lower right-hand corner if you're interested. So a starfish actually has over 1 million pieces to its body, 1 million plates, these little bitty things. So when we look at a fossil like this, it's actually an incredibly rare event that we would find something that looks this beautifully preserved, right? And we can take a look at a less familiar echinoderm to us. Take a look at this. This is a Diploporiton echinoderm called Eumorphocystis. All right, it's a species name, or genus and species name, excuse me. And while this likely has a few less plates than the starfish, it still has quite a few plates, and this is absolutely beautifully preserved. Well, this isn't what most echinoderm fossils look like. All right, so let's take a look at this graph. This was published in Brett et al. 1997, a very famous paper for this. We can see that Brett has broken down echinoderm taphonomy into 3 types, type 1, 2, and 3. Type 1 are things like ophiuroids, the brittle stars, asteroids, the starfish, things like that. And you see that after 1 day, usually, the pieces of a starfish are broken down to a point where they're almost unrecognizable. And after a year, well, they're broken down into absolutely nothing, right? These little tiny unrecognizable bits. Now consider that the starfish that I just showed you is over 300 million years old. I think closer to 400 million years old. So these are incredibly rare fossils to find. Now, I'm not working with things that live in type, or that are type 1 specifically. I'm working with type 2, which are slightly more robust, meaning they hold together a little bit better in the fossil record, but not much. So examples of this are things like crinoids, the sea lilies, echinoids, the sea biscuits, or the group we're going to be focusing on, the cystoids. You'll notice cystoid is in quotation marks. That's because it's a polyphyletic group, all right, meaning that it has multiple groups kind of shoved into one, so it's not a real scientific term. So here is an example of a cystoid. This is a rhombiferan, and how it gets its name is if you'll notice the diamond-shaped patterns that you're looking at, those are called the rhombs. That's where it gets its name from. But what you'll notice here is this is a reconstruction of an organism that would have been perfectly preserved in life. But in the fossil record, we very rarely see the stem pieces. And we also very rarely see the brachial pieces. And the brachials look like, they're the things that look like arms on this specimen. And when we're missing both the stem and the brachial pieces, we're limiting what we can understand about the morphology of these groups. So that can be a huge problem when we're trying to assess evolutionary patterns. So let's take a look at some other fossils, all right? So here we have 2 different specimens here. One is clearly much better preserved than the other. If you take a look at the one on the left and the top there, that's not so well preserved, right? It's kind of ugly, if I may say so, call a fossil ugly. And we take a look at the bottom and the one on the right. You see that it's beautifully preserved. Look at all of the plates and all of the features that have been preserved, but you're still missing all of those brachial arm pieces that you saw on the previous specimen. These specimens have it too, but they're missing, right? So it's very rare that we actually find a lot of these pieces. So we're going to take a look at some of the fossils that are going to focus our talk today. So these are called the blastozoans. And the blastozoans mean nothing more than echinoderms with those special brachial plates that look like arms, and they're for feeding. And they also have specialized respiratory structures for breathing. So for an example of this, one major group here is called the Diploporita. And that simply means that it has diplopore respiratory structures. Diplopores are double pores. So if you look at the body of this fossil here, again, this is Eumorphocystus, one that we've seen before, you see that there's little tiny holes all over the body here. And what it's doing is these are the respiratory structures. This is how the organism breathes. There's 2 pores per set, and gas exchange occurs across these pores. So here we have an up-close image of a diplopore. You can see that those 2 holes are sitting within kind of a depression all over the body. This is Haplospheronis. Some of them can look a little different, right? So here we have a humatopore, diplopore, and what you see here is we still have that 2-hole set, but it's actually connected by a bunch of different tubes. That's the only major difference between them, just so you know. So we're going to focus our talk today on the Holocystides fauna, which is an incredibly unusual group that shows up primarily in North America in the late Ordovician to Middle Silurian. All right, and here's a beautiful photo of one of them, Holocystides spangleri. All right, so here is an example of the side piece on the left, or the side view of the body. In the middle, we have the oral view looking down at the mouth. And on the right, we have an example, a line drawing of what that oral area looks like. So we're going to talk about what makes a Holocystides fauna the Holocystides fauna. So first, it's defined by having humatopore respiratory structures. So all of them have that special type of diplopore that's connected by a bunch of different tubes. We have a holdfast. There's no stem in this. We have a holdfast. And what that holdfast does is it secretes a type of mucus that allows it to stick on things like other shells or on hard sediment, something like that. Has a relatively large body size. All of these creatures have evolved very large bodies. And it has 7 oral plates surrounded by 7 facetal plates. So if you take a look at the line drawing, you see the oral plates are outlined in gray, and they're marked things like 1, 2, 3, 4. All right, and then we have the facetal plates, which are outlined in white, and they're A, B, C, D, E, and the 2 L plates. All right, those are the facetal plates. And these plates border a large peristome. The peristome can simply be called the mouth, so it's labeled by an M directly in the center. You'll see opposite O7, the oral plate 7, you have a P, that's the periproct or the anus. All right, so there we had the oral plates, and now we have the facetal plates highlighted, just so you can see it a little bit better. They also have very large brachial facets. And again, brachials are those plates that look like the arms coming off the body. So I've highlighted the brachial plates in these yellow arrows, and you can see that they're relatively large brachial facets, almost like a scar left behind. And they're attached to short ambulacral grooves. And ambulacra are the food grooves. So the taphonomic pattern of Holocystids is pretty simple to figure out. All right, so let's start talking about the one on the right first. You'll notice that unlike the one on the left, where we have clearly labeled 0 or 0, 1, 2, 3, 4, 5, and 6, you actually only see little tiny bits of those oral plates and O7. That's because it's incredibly common for these oral plates to fall out very quickly after death. But what you may not know is that covering M and P, again the mouth and the periproct, the anus, there's also a series of plates that should be covering it. The primary peristomial cover plates should be covering M, and we have a anal pyramid covering P. And those are almost always disarticulated. So here just showing you again, there should be a series of plates covering M and a series of plates covering P. And you can see both M and P there in the real specimen on the right. However, new specimens from Silurian, the Silurian Massey Formation, which is found in southeastern Indiana, and here's a map of showing you where this is in southeastern Indiana, indicate that we have new and unusually well-preserved specimens that show us some of these plates that we rarely see. And what's special about this area, this Massey Formation, this area in southeastern Indiana, is that there are hundreds of specimens of these diploporitan fossils. And this is unusual because most places in the world don't have these fossils preserved in a very high quantity. Most are very low, very few specimens per area. This has hundreds of specimens, many of which are well preserved. So it's a fairly unusual and wonderful area for fossils. So here we have an example of a rare specimen showing those preserved primary peristomial cover plating, the PPCP, if you wanted to say that for short. So what you see here covering that mouth, the M, is we have a few large cover plates right? You see 5 cover plates there. So what you notice here is that they are very large, and that's important, and I'll show you why in just a slide. And you'll notice that these plates lack any indentations across them, and what this can tell us is it can separate us from other diploporitan groups, which have slightly different features preserved in these cover plate systems. So taking a look here, we have on the left our Holicysteides that we've been looking at for a little bit, and on the right we have Eumorphocystis, and this is the oral area view, the top view of Eumorphocystis. And you'll see the PPCP, the peristomial cover plates, preserved, and they're colored in as dark blue, but you see there's many of them, and they're very small and separated out from one another. So what this can show us is exactly how different these two fossils are in the types of cover plates that they had. So we have more information concerning their evolutionary history now. And if you take a look at this, we have the anal cover plates preserved in this one particular specimen here, so that's indicated by the yellow arrow. And what we know here is that these cover plates are actually much more conserved across blastozoan clades, and we have that information now with this particular fossil. So with this Holicysteides, we can see that these anal pyramid plates look very similar to other blastozoan clades or groups. And here's just a close-up view of it. All right, you see that here we have that anal pyramid there, and it's in contact with the oral plate number 7, which is directly above it, just like you saw in those line drawings before. And this is one of the most beautiful and rare fossils that have been found from this location. What this is an example of a preserved brachial, that arm-looking structure that we've been looking at. So what we know here is it's uniserial, meaning that these plate series aren't divided into 2, which would be called biserial. They're proportionally very large compared with other examples of blastozoan brachials, and they extend quite some distance from the theca, quite a few centimeters, in fact. And this is what we think it's closely related to. We believe that this specimen, shown here again on the left, is in the same group as this one here on the right, Triamera, which is just a genus name. The species here shown is Triamera ventricosa. This is not Triamera ventricosa likely, it could be another species, but it belongs into the same group as Triamera most likely. And what you see here, yellow, indicated by the yellow arrow there, is that where that brachial plates would have attached to. There's the attachment scar right there. And why this is so unusual is that these brachials that have been found are incredibly dissimilar to other blastozoan brachials and even other diploporitan brachials. And remember, a diploporitan is a type of blastozoan. So if you take a look at the yellow arrows here, I've highlighted both of the brachial facet scars. And you can see that proportionally, the ones on Triamera, the one on the left, are much larger than the ones on the right. And the one on the right is called Astonocystis. It's found in Estonia primarily. And again, this is very dissimilar to other blastozoans that aren't diploporitans, like these rhombiferans, right? This is one we saw earlier, Lipsanocystis. All right, and you can see that there's many of these brachial facets that would have been attached in life, and they're quite tiny. So let's take a look at this phylogenetic tree that's been made very recently. This is work that is being submitted currently to a journal for publication by Sheffield and Sumrall. And what we see here is that Diploporita is a polyphyletic assemblage. I've highlighted all of the groups that have been traditionally named within Diploporita. So we have the Spheronotida, which is in red, the Glyptospheratida, which is in yellow, the Astroblastida, which is in blue, and non-diploporitan echinoderms, highlighted in green. And polyphyletic assemblage means that the group doesn't have one distinct ancestor and the descendants are linked together, but it means that the descendants are actually scattered throughout the tree, which is what we're seeing here. You can see that green shows up in the middle of the yellow, you can see yellow showing up in the middle of green, blue's all the way out without not even close to the other diploporitans. This is a polyphyletic assemblage. All right, the only monophyletic group that we've uncovered is in the red, the Spheronotida, which is where the Holicysteides fauna, the group that we've been talking about, can be found. So what we see here are some interesting patterns with the evolution of these brachials. So if we look at that clade, the Spheronotida, right, the monophyletic clade, we see that all of these examples have short ambulacral food grooves that are restricted to the oral area, meaning the top of the specimen. And you can see that across all of these specimens outlined here at the top of the tree. They have very short ambulacral food grooves that are restricted to the oral area. Within this, there are two other groups though. One, ambulacra that end in multiple single terminal small brachial facets that almost look like a little fan coming out from the mouth of the specimen. And the other group, which has ambulacra that end in large single terminal brachial facets. All right, so you can see here that there's only one facet per ambulacra. That's a very big difference, and I'll show you that again. You see here where there's multiple of them versus one of them. And finally, we look at this other group. These are the glyptospheridids, which do not make a monophyletic group. These ambulacra extend down the theca, which is just the body, with alternating small brachial facets that look almost like little dangly Christmas lights, if you will. And finally, with this astroblastid, we have ambulacra that are recumbent, meaning laying down against the body wall with bilateral brachial facets. You see how if you look at that groove there, there's facets on either side, almost symmetrical. So you see that there's major changes in the makeup of these brachials. All right, even if you don't understand the terminology necessarily, the important part is you can see the changes in the shape. So the question here is, why do we have this change from very small brachials and multiple brachials per ambulacrum, those food grooves, which would, if you will, I mean, these are for feeding, right? So the more that you have, presumably, the better you have, the better structures you have for feeding. So why would we have this change from very small brachials and multiple brachials per ambulacrum, as opposed to what we see in the Holeocystides fauna, which is one large brachial per ambulacrum? Well, that's what the direction of our future work is taking. So to assess the evolutionary trends in these brachials, we are looking to quantify the morphologic disparity, meaning the amount of diversity in the shapes across these groups, the spheronotid diploparitans. And again, remember, the spheronatids are just that monophyletic group that includes the Holeocystides fauna that we were talking about. And when we do this, we're going to try and assess exactly why these changes are happening. So what we know is that in the late Ordovician, a time period, we had a mass extinction, a glaciation that was responsible for causing the extinction of most of the diploparitans, except the Holeocystides fauna. So what we see is one single group coming across the Ordovician to Silurian boundary and then starting to speciate more rapidly across that boundary with these large ambulacra that only have one brachial facet. So the question is exactly what is allowing for that to be a successful trend? So if you take a look at this graph on the right here by DeLine, 2015, this is quantifying morphologic disparity, essentially, in kind of layman's terms here, how different each of these groups are from one another. All right, the Diploparita is highlighted in a dark green color, kind of in the center of this graph. And you see that that dark green color is very widely spread across this, meaning that the Diploparita themselves are very different from one another. All right, so what we're trying to do is assess why that difference exists. Could it be because of certain climatic factors? Could we tie this to climate change during the Ordovician and Silurian? That remains to be seen. So far, I would like to thank the funding sources that allowed me to visit these museums to see these fossils, and I would like to thank especially Don Bissett of the Dry Dredgers Fossil Association in Ohio for collecting these new beautiful fossils. And I would like to thank all of the museum curators that provided the other specimens that were used in this analysis, from the University of Iowa, from the Chicago Field Museum of Natural History, from the Geological Institute of Tallinn in Estonia, the Yale Peabody Museum, and the United States National Museum in Washington, D.C. And with that, I'd like to thank you very much for listening. Bye.